Hello everybody. So in last lecture I stopped here. We were talking about the motion perception, and we talked about the motion parallax, optic flow, those important clues or cues for visual motion perception. uh, Here in this slide you can see a motion illusion. So a lot of those reasons of many of those illusions are not clear. We don't know the exact neural or brain mechanisms for the visual illusions. Multiple areas are involved in creating those illusions. Uh, so here you see that one kind of illusion, very popular. This is called Auchi apparent motion. If you see the left side's circle, you see the white and dark bars are there, and in the central disk of this circle you see the bars are in opposite direction. And if you look at the center part uh, of this circle, put your eyes on that disk, you can see that it gives you a perception that it is moving. So that is an illusion, and it is suggested that that illusion is due to high contrast of the color. Black and white, they're in highest contrast form, and also the direction of those bars are in opposite direction. But if you see the right side circle, you don't see that motion illusion because of lack of contrast. So that is one type of visual illusion, and we, as I mentioned, that we don't know the exact mechanisms of those. There are some hypotheses, but still it is not clearly known. Motion after effect. If you watch at put your eyes in the center of this moving circles for a while, and then if you move stop that motion, what you will see that you will feel that the circles are going uh, in opposite direction. Now you see that circles are coming towards you, opening towards you, or expanding towards you. But if I stop that, it gives the perception that uh, all those circles are going backwards. And another thing, after you look at this motion, then if you suddenly move your eyes uh, on something else, for example, if you move your eyes to the text above that, you can feel that the size of those letters are getting smaller, the text is getting smaller. That means moving away. So that is called motion after effect. If you look at the waterfall for a while, and if you suddenly move your eyes to the trees or something else, you can see that everything is moving upwards, because the fall you are looking at moves the waterfall, water moves downwards. So that is called the motion after effect. You can, when you look at something else, the things move to opposite direction. As I mentioned already, that the scientists have suggested that many areas are involved, many different types of direction sensitive neurons in different areas of the brain uh, could be responsible for that. This is another type of motion that is called biological motion. Uh, you know, if you see the camouflage, you know that it is difficult to um, identify the object or living object if it is, in camouflage in a forest, but the animals detect the prey by looking at the movement. Even the color is similar to the background. Uh, if the color is similar to the background, it is difficult if the object is in a static or non-motion state. But even if the color is same, the object and the background, if the object moves, then it is very easy to detect it. So that is one type of motion that is called biological motion. And human brain is very good in detecting biological motion, as well as many the predators, the creatures. Those are those live by hunting or catching prey. Their survival depends 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 on that. So that biological motion is very important for certain creatures live in the forest. Human brain is also good, as you see that only with few dots you can clearly tell what is happening here, right? But if you see these dots, I show you these dots, you won't be able to tell what is going on here. But if you see here, uh, someone can just quickly tell that a person is moving, walking, right? So that is the biological motion. Structure from motion. This is another example of motion that you can create by the motion of the object. So here you can see that a number, two different colored dots, white and black, and they are moving in opposite direction. You can see that, and by this motion or movement, you can see that how a beautiful cylinder is being created. A moving cylinder is being created. So this is a good example of structure from motion. Motion is a very important uh, clue for the structure. Okay, so those are a few visual illusion, and those are related to the motion, object motion. Now you can see how the neurons of V4, area V4, can give you the perception of a shape of a large object. As you know that when we talked about V1 or primary visual cortex, you have learned that the size of the receptive field is pretty small, because that is the earliest visual area in the brain, and the size of the receptive field is much smaller than V4. And as the signal goes upwards, the size of the receptive field of the neurons get bigger. So V1 neuron, you must remember that are good for detecting the edge, particularly simple neurons of V1. They are good in detecting the edge, but there is, since the receptive field size is small, they cannot give you the perception of a total picture. So here V4 neuron size is much bigger, and you see a number of V4 neurons, um, the activity of a number of V4 neurons have been shown here. And if you see if you see this area, these neurons are sensitive to this convex curve, and B and D are processing the concave surfaces. C is very strongly sensitive. The neurons of this area are strongly sensitive to this sharp, pointed angle. And if you see F, these neurons here are sensitive to this flat kind of convex surface. Okay, so you see these groups of neurons are working together, V4 neurons, to give you the perception of this shape. Okay, so that's how V4 neurons work together and help you to give the perception of the shape of an object. V4 neurons, I mentioned before, are also responsible for uh, color, processing of color. So these are two important functions of V4 neurons: shape and color perception. Okay. Recognition of face and object. Face recognition is a very interesting topic. If you can scan the movement of your eyeballs or eyes while you watch someone's face, look at someone's face, it is very interesting that you will find uh, the eyes of the observer spend a lot of time in particular areas. For example, in the lip, uh, on the eyes, nose. So the eyes move like this, and at same location scans for longer time. So more time is spent on those particular areas of the face when you look at someone's face. And this is something interesting. Also, hair, the edge of the hair. So that saccade and microsaccade both are important, and the microsaccade that scans the particular area uh, helps you to refresh your retinal image. So refreshing the retinal image to keep the signal going continuously towards the brain. That is very important. If you inject lidocaine, for example, which is a local anesthetic to paralyze the muscles of the eye. So eye movement, if you can stop the eye movement, those movements of the eyeballs, then the things get gradually less visible. The reason is the retinal refreshment stops. So retinal refreshment, or the small movement of the retina is continuously helping to refresh the image and sending signal to the brain. Uh, to recognize the face, you have particular areas in the brain, in the uh, temporal part of the brain, which is the anterior or ventral stream. So the most powerful area for the recognition of face is called fusiform face area, FFA. And this area, fusiform face area, if you see that area, inside that area you have discrete neurons, very highly concentrated neurons. Those are sensitive to face. So these neurons produce action potential or get excited or fire when you see someone's face. So face recognition is the main function of the area FFA, fusiform face area. But some face sensitive neurons are found in other areas too, but they are scattered. But in fusiform face area, you have huge number of face sensitive neurons there. This is one of the earlier experiments where the fusiform face area was investigated on monkey's brain, and you can see that when the subject was a monkey, and when the monkey looks at a monkey face on the screen, the neurons of fusiform face area responds very strongly. This is the experimenter. So monkey knows his face, and when you show the monkey the experimenter's face, you see the firing of the neuron of that area increased. This response is stronger than this one, I would say. And now you see something interesting. If you erase the eyes and nose, particularly here, you see they have removed the eyes from the picture, so the firing decreased a lot. If you show the monkey a cartoon face emoji, you see the firing is totally gone. That means spontaneous firing is there, but the response is gone. Firing is still there, but at a spontaneous rate, but the responsiveness is not there. And if you show scrambled picture, no signal is there, no face signal is there. Its firing is pretty low and no response at all. So this clearly indicates that fusiform face area neurons are very sensitive to face recognition. This is actually another experiment was done by a Japanese scientist Matsumura. I actually worked with him for a while in Japan. He works at Kyoto University in Japan, and his name is Matsumura, Kyoto University. And here you see that same kind of experiment. When the monkey saw a monkey face, the response of the neuron is very strong. If you see the scrambled, no signal is there, spontaneous rate. Here, when the monkey sees the experimenter's face, see the response is pretty strong. When the monkey sees the picture of a hand, the response is low. Uh, Again, this is an experiment where the recording was performed by fMRI in human infero-temporal cortex. So in this recording, you see they used a number of objects, human face, then other objects. So here you see the activation of the infero-temporal cortex recorded by fMRI, and here you see the object, either of these three, and then face, then another object, face, face. So only you see the when the subject, human subject, saw another person's face or human face, you see the response of that area increased, activity of that area increased only when the person watched the face, human face. They didn't use the same face, they used different faces, but same response they observed in all cases. But when the subject watched the objects, other objects, you see the response is pretty almost no response. Okay, white bars. Here you see the experiment was done by using human faces again and houses. So you see this fusiform infratemporal cortex area of human only responds when you show the subject human face. When you show the houses, no response. Okay, so face, house, face, house. And in this case you see the subject was shown human faces again and scrambled faces. So when the subject watched the scrambled face, yes, right, no response. When the subject watched human face, you see the response is pretty strong, right? So uh, this experiment clearly indicates that the area only prefers the face, not any other object, not other objects or houses or scrambled face. So we have, like monkeys, human brain also has face area that is responsible for the processing of the face. Here plays an important role to give you the perception of face. You see here the same person's face, but if you change the hair, the look is very different, right? So these two faces are same, George Washington, but if you give him Elvis Presley's hair, he looks very different than the president. So you can see George W. Bush, and he got Elvis hair style, so the face looks different. So that's why when the robbers, they do bank robbery or they do robbery, they use the uh, monkey cap or cap to cover the face, cover the hair. Orientation is also a factor that is related to your perception of face. For example, same picture, if you put in right orientation, that is the orientation we used to see when we see someone's face, uh, you can clearly tell that something is wrong here and here, right? So you can see the odd uh, very quickly. You can detect that. But if you change the orientation, move it upside down, uh, we are not used to see the face this way, so it is difficult for us to detect the odd areas or odd. So orientation, or the orientation that we are used to, gives you better perception. Okay, so now you know that for face area, for to recognize the face, both monkeys and human subjects have certain areas in the brain, in the ventral stem or temporal uh, lobe areas. Now we will talk about the object recognition. Non-face object recognition is one thing. So in IT, the neurons are present in columnar organization, and IT has a columnar organization, and if you record the neurons within the same column, you will see the orientation of the object uh, is perceived by those neurons in same orientation. For example, this is the side view of a face, so all these neurons responds to this orientation. Now if you record the neurons from this column of IT, you will see that all neurons are sensitive to this orientation of face, or this uh, image. If you record here in this column, different, but within the column the neurons responds to same uh, stimulus. Same thing here. If these neurons like this shape, all these neurons would respond to this type of, or this shaped stimulus, and these neurons would respond to this shaped stimulus, and these neurons in this column would respond to this shaped stimulus. So in IT you have the columnar organization, and within a column the neurons are sensitive to same kind of shape. Now what is the difference between the neurons of earlier visual areas, for example V1 and IT? We know that IT is in ventral stem, right? You remember IT is in ventral stream, and ventral stream is responsible for which is also called shape, hot area. So shape is an important cue for identifying the object. So shape, color, those are processed in the ventral stream, which is the temporal uh, lobe. We talked about that before. Okay, so if you record the neurons from infero-temporal cortex, the cells respond selectively to a particular class of object, for example, faces, hands, animals, etc. Cells that respond to the shape of hand do not respond to faces, that we talked in last slide, and cells are tuned to particular instances of object, for example, particular animal. And the response of the cell is the same independent of the location. So these IT neurons, inferior temporal cortical neurons, doesn't care the location because this is in hot stream, not where. So it doesn't matter where the object is in space. It only cares the shape. Okay, it doesn't care the size. It only cares the shape. So the neuron will respond same way, even if it is here, here, or here. It doesn't matter where the object is. And also the neuron would respond same way if you change the size of the object. As long as the shape remains same, the neurons will like that and will respond to that particular shape. It's invariant of the location or size. Okay, so location, size, as well as cue. You can change the color or you can change the texture, doesn't matter. The IT only cares the shape. So these shapes are all same object, right? So that is something important to know. If you record the neurons of V1, you will see they're totally different, right? The response changes with the change of the size. Okay, V1 neurons prefer particular size, so different neurons prefer different sizes. So if you change the size, the response will change. If you change the location, if you change the color, the response will change. And in V4, definitely, you already know that responsible for color of the object. So if you change the color, the V4 neuron will show the change of response. So difference between V1 and IT cells responses. V1 won't care the shape. So as long as the size and color, those are same, one V1 neuron, the same V1 neuron will respond to this as well as this, because the size and the color, those two things are uh, you know processed in V1. If you see IT, neurons of IT, the neurons will respond same way to this one and this one. It will not change the responsiveness because of the change of size or color. Whatever the color or size, as long as the shapes are same, uh, the neuron will respond same way. Okay, so that is the difference between the V1 and IT. Visual attention. Attention is very important in our everyday life and related to many things. You know that attention uh, or focus, if you pay attention or focus on something, you learn better, right? It takes shorter time to learn. Reaction time to respond to anything is faster. Quick response. Uh, so those are just uh, attention can change those things, can change the response time or reaction time. So if you pay more attention, reaction time will be shorter, decreases the reaction time and increases the response, correctness, correct response, and also increases learning. You know that if you pay more attention, you learn faster and better. Okay, memorize longer. So those are just few things. Those are very much related to attention. Now, uh, the attention is controlled by the higher visual areas, and higher areas control the lower level visual functions. This is a simple example, like when you see, where if I ask you to read a sentence where you have this word, many of you will miss this and will, without noticing this, error here. This should be M, but many of you will read it example if you don't pay attention. Now, if you pay attention more, you will be able to detect this error, okay, or typo. So from where the attention comes? Attention comes from the higher areas. And memory also plays an important role here. If you, this is the first time you have seen this word, this is a totally new word for you, then of course it will be difficult for you to correct it, right? So memory creates a template in the brain, and when we see the same thing, your brain quickly matches that object uh, to your template and gives you that this is an apple or this is an orange or something, right? So you already have a template in the brain, that is the memory, and when you see something, goes through the visual cortex and your brain interprets based on the memory that you have. Okay, so that is one thing. Memories come from the higher brain areas uh, to the interpretation area. Okay, so that is an example that how the higher areas are controlling the lower areas. Uh, if I ask you quickly that what is the color of this, some of you will say red. because that's a contradiction. You see the color, between color and the word that you see. So two different things are being processed in the brain, and they contradict each other, so that for a while you will get confused. If I ask you to tell me what's the color, some of you will answer blue, some of you will answer red, but there's a good chance that some of you will answer blue. So paying attention comes from the higher areas. Now if you pay more attention, then you will be able to answer more correctly. Okay. um, eye movement. We see different types of eye movement. One is of course voluntary eye movement. If I ask you to follow something, when you see a ball is approaching towards you, towards your eyes, uh, then what happens? You, as the ball moves, you keep following the ball. This is voluntary eye movement, right? If I ask you to see an aeroplane or a car, moving car, you follow your eyes. Your eyes follow together because they are connected, two eyes, so they must move together. So that is voluntary eye movement. Uh, Involuntary eye movement, when you involuntarily your eyes move towards something. If suddenly a light flashes, sometimes automatically the eyes move towards that. Now the movement could be voluntary and involuntary, but also we classify the eye movement uh, another way, that is pursuit and smooth pursuit. So when you are asked to follow something, you move your eyes slowly, that is the pursuit. And saccade is different than pursuit. Saccade is sudden movement from one object to another. So if I ask you to move your eyes from this object to this one, you move your eyes suddenly from here to here. So that is the saccade. Okay, and microsaccade is, that I talked about that before. When you are scanning someone's face, your eyes are moving like this, that is microsaccade. Okay, so pursuit, constant following, and saccade could be uh, saccade, the first movement from one location to another, and microsaccade is small movement. As I have mentioned that attention, visual attention is very important because it improves your ability to detect something, discriminate the things, um, and shortens the reaction time, and plays significant role in learning and memory. Okay, so many experiments have been performed on the attention. Psychologists as well as neuroscientists performed many experiments. In neuroscience, we depend or rely on the brain function, but psychologists, they don't rely on the neural mechanism much. They just create the hypothesis. Anyway, so there are different ways to do the attention experiments. Attention could be endogenous attention uh, and exogenous attention. Okay, so what is the difference between these two, exogenous and endogenous? Uh, Endogenous attention is like if I ask you that in this PowerPoint slide, um, something will appear and you have to quickly respond when you see it. Okay, now if I tell you that that object will appear in the upper right quadrant of this, whole visual field, you will pay attention to this upper right quadrant of the visual field. You will not pay attention in other areas. So when the object will appear, since you know it before, your higher brain areas will you know keep you alert. Your recognition areas will stay alert, and because they are getting signal from the higher areas, uh, and when something will appear here, you will be able to quickly respond. Now if you don't have that clue, if you know that it can appear anywhere in the visual field, then you your focus is not as much as in this case. Okay, so in that case the reaction time or the ability to respond will be slower. Okay, so that is the endogenous attention. You already have some information, okay, preassumption about the location or about the object. Okay, uh, exogenous, if something happens outside, like while you are driving a car, if you listen the sound of the traffic car, police car, petrol police car, you suddenly pay attention more, right? So that is coming from outside. You did not have any pre-assumption or previous information about that uh, stimulus or signal. That is totally coming from outside. So that is exogenous. So uh, now exogenous is bottom up because coming from outside, and first the eyes or ears, whatever the peripheral organs are receiving the signal and sending towards the brain. Okay, then you pay more attention. So this is bottom up, but in this case it is top down because your higher brain areas already knew that something will appear in this particular area of the space, right? So it is top down, and this is bottom up. So we know that there are two streams from V1. Signal can go to dorsal or ventral streams. That means where and hot streams, right? We talked about that a lot. Okay, and attention could be either, right? Where the object is appearing, that is processed in this area, and if the uh, question is what is that object, that is processed in this area. So both areas are related to attention. Now I told you that higher brain areas control the lower areas. So what is that higher area? Prefrontal cortex is the main higher areas that have the uh, presumption or information about the cue. some information about the stimulus, so it will help to identify the object quickly. So here you see the prefrontal cortex. Now you see that from the eyes, visual cortex, primary visual cortex, then from there signal can go to either posterior if it is where, location related attention, or hot, uh, the object related attention, the identification of the object related attention. It can go to either pathways, and then from there the signal will go to the prefrontal cortex, and prefrontal cortex then can send signal to the superior colliculus to move the eye to that object, right? Uh, and also send signal to the memory and recognition if uh, your brain needs to keep that information as a template or as stored in the memory. So some information will go to the memory system of the brain. Every time you see something and pay attention, signal will also go to the memory areas of the brain to store uh, that information. So if you see the same kind again, then you can quickly match with the template that I talked before. And you also move your eyes to pay attention uh, to that object, and also signal goes to the motor and other uh, areas. Now here you see what I mentioned before, that from the prefrontal cortex, the higher areas, signal goes down to those areas. So if you have previous information stored here, then signal will go to those areas and to make these areas more efficient. Okay, so that is something very important function of the higher area. Uh, you already know the functions of dorsal pathway and ventral pathway. Uh, Damage to the dorsal pathway can lead to a clinical condition that is called optic ataxia. The subject would have difficulty reaching for and grasping the object, uh, and this often results due to damage to the superior portion of the parietal cortex, okay, or parietal lobe. And the patient can recognize the object because this is a damage or lesion to the dorsal stream or dorsal pathway. So object recognition will not be hampered. It will remain, functional, because we know that object recognition hot pathway is in the temporal lobe, not in the parietal lobe. So the subject will be able to recognize and describe the object, the shape of it, and what is that. The problem occurs in the where pathway, where function. So the subject will not be able to recognize the location and will not be able to precisely reach to that object or grasp it. Now if damage occurs in the ventral or hot pathway, the clinical condition is called agnosia or anoxia, and patient can accurately reach the object and grasp it, uh, but the subject has problem in identifying the object. That makes sense. this is an experiment was performed on monkeys LIP, and we already know that LIP, lateral infero-temporal parietal. So this is parietal, that means dorsal stream, and the neurons of LIP reflects intended eye movement. And how you know that? The experiment, this is a nice experiment. You record from LIP neurons, and first T1 at time 1, the monkey will fixate eyes on the center of the screen, computer screen. So that is the fixation of the eyes, and monkey will maintain the fixation until it receives next command. So we'll keep fixating eyes on this center point, that is the fixation point, and this is the receptive field of that recording neuron here. And you see at time, this is time 0, t0. So first the monkey will fixate the eyes, and you measure the receptive field location of the neuron. Then the monkey will see, while fixating his eyes, within the receptive field an object, this red circle, will appear, and monkey was trained to first see, and while it is seeing that red object, uh, it must keep fixating and think where the object is. And then the monkey at T2, monkey is planning, and when at T2 that red object here will disappear. So monkey is planning, thinking to move his eyes, and the object inside the receptive field disappears. And then once the object disappears, monkey is supposed to move the eyes from the fixation point to the object. Now every time you move your eyes, that means the receptive field of the neuron moves same way, right? So if you move the fixation point from here to here, your eyes are moving from here to here, the receptive field of that recording neuron will also move that way, because receptive field is also maintains the same distance from the fixation point, from the fovea. So now let's see, this is T3 when the monkey moves the eyes to that target. So this is T0, T1, and T2 is here. So this is T1, this is T2. So you see, when the target point appears during T1, at T1, the firing increased a lot. That means when the monkey was intending to move his eyes, thinking where to move the eyes, during that time, although the eyes are still there, the firing increased while the monkey was thinking to about the location where it has to move. Before it moves, the firing increased a lot. Okay. Then between T2 and T3, you see here, between this and this, that means before the eyes moves, the firing is not that high, but still higher, much higher than the baseline. So this is the baseline, so this is much higher. So that means the monkey is still thinking where to move even after the target disappeared at T2, and before the monkey moves his eyes, the firing continued to stay um, higher than the baseline. So that means as long as the monkey is intending to move the eyes, paying attention here, its firing rate is much higher than the baseline. So that tells that LIP neurons are uh, processing that intended eye movement information or signal. This is an experiment of attention. Shift of attention affects neural responses of V4. So V4 neurons are also responsible for attention. So many brain areas are related to attention. This is an experiment where you see that this is the fixation point in the center of the screen, and this is the receptive field of V4 here, and you see the color. What happens, the color of the fixation point changes and two sets of bars appear on the screen around the fixation point. One is red colored and another set is blue colored, and uh, the monkey was trained to pay attention to the color. When the color of the fixation point changes, uh, then monkey has to pay attention to that particular colored uh, stimulus. That means the same colored lines. So now you see here, this fixation point color is red, so monkey pays attention to these bars or lines, and these are outside of the receptive field. Now the color of the fixation point turned to blue. So now the monkey, while fixating eyes here, since the color is blue now, the monkey will pay attention to the blue bars. And the blue bar, one blue bar is inside the receptive field. So in this condition, this neuron is paying attention to this. So blue. So the firing will increase a lot. So you will see that here. And in this side, you see this is another condition. In condition 1, attention shifts from outside receptive field to inside. First, the attention was on the red bars, so outside of the receptive field, then moved to inside the receptive field, to the blue line. Here, from inside to outside. That means the blue fixation point. So monkey first pays attention to the blue, and blue is inside, and then the blue fixation point turned to red, so the targets were outside of the receptive field. So that's why you see opposite. This is condition 1, right? So in condition 1, in first part here, in this case, the attention was outside of the receptive field, right, on the red bars, so the response was very low. And when the fixation when the attention was moved to inside the receptive field of that neuron, the neuron responded a lot. Okay, so attention increased the responsiveness or response of the V4 neuron. And this is opposite. First the attention was inside, so the first part you see the response was very high. Then when the attention was paid outside of the receptive field of that neuron is not uh, responding. So this is a nice way to demonstrate that how the attention changes the responsiveness of area V4 or the neurons of V4. Okay. cue, if the cue is given earlier before the task, that can improve the uh, perception significantly. We all know that. Like, this is time 1. The subject fixates eyes on the center, to the center of the screen, and at time 2 the cue will appear. So uh, this is the cue, and in this case 80% of the trial got valid cue. That means what? First the subject was given this cue that the target will appear to this side of this arrow. Okay, and in 80% cases the subject was given the valid cue. That means when the subject first was shown this direction, and in 80% trials the object appeared in that direction, and in 20% cases or trials the subject was given the cue this leftwards, but the object appeared in the other side. So the subject did not have any had wrong information. So this is invalid cue, right? So this is invalid cue. This is valid cue because this was the right direction to the object. Okay, and then this one, when the subject was shown this doesn't carry any signal. It doesn't gives any information if the object will appear in the right or left. So 50% cases it was object appeared in the left side, 50% in the right side. So this is called neutral cue. And here this is again same way, this, but just opposite direction. The cue was given that the object will appear in this side, and in 80% cases it happened. So this is valid cue. The reaction time would be very fast, right? Because already you know from this one that the object will appear here. So when the object appeared here, the subject quickly moved the eye or detected that. Okay, but if you are given the cue that object will come this way, but when the object appeared, it appeared in this side, then it took longer time, even longer than the neutral cue. So here, this is the reaction time or response time was much longer. In case of invalid cue, it was less than invalid cue, um, and was very short for the valid cues because you already had information. Okay, so also the accuracy of response increases if by the valid cue. So two things, as I have mentioned earlier, reaction time gets shorter, and correct response increases. Okay, so um, so valid cue increases the chance of correct response and decreases the reaction time. That means performance improves. So let's stop here. So you have learned some important things about the attention, shape, and face recognition, and the areas those could be involved in that.